Bismillahirrahmanirrahim. Previous lecture discussed the transcriptional process for the synthesis of RNA. And RNA, it comprises ribose, phosphates, and the nitrogenous bases. These are the basic components of the RNA. Ribose is the ribose sugar instead of the deoxyribose, phosphate which is utilized for the diester linkage, and the four types of nitrogenous bases: adenine, uracil, cytosine, and guanine. Instead of thymine, uracil is present in the RNA, and all of these components they are utilized for the formation of ribonucleic acid. There are different types of ribonucleic acid: ribosomal RNA, tRNA, and the messenger RNA. Ribosomal RNA, they are destined for the translational machinery. They associate with different types of proteins, both in prokaryotes as well as in eukaryotes, so as to form a ribosome. Now this ribosome, they are the translation machinery which are utilized for the formation of proteins. There are three distinct size species of ribosomal RNA in case of prokaryotes, but in case of eukaryotes, the four size species are present, that is 28S, 18S, 5.8S, and 5S, while in prokaryotes the 23S, 16S, and 5S ribosomal RNA, they are present. These ribosomal RNA, in case of a prokaryote, they are formed from a single polymerase enzyme, but in case of eukaryotes, the 5S ribosomal RNA, it is synthesized from polymerase 3, while the RNA polymerase 1, it synthesizes the other three species of ribosomal RNA. Now next is the tRNA. The tRNA, it is transfer RNA. It acts as an adapter molecule to carry the specific standard amino acid to the codon present on the messenger RNA. So it gives the link for carrying and bringing the specific amino acid to the codon for the synthesis of polypeptide chain, and also it assists, it help in the translational process. The tRNA, the characteristic feature of tRNA is that it contains the unusual bases, extensive intra-chain base pairing present in its structure, and it also undergoes the different modification um, so as to carry its proper functioning. Along with there is the presence of the CCA nucleotide at the 3' end, which carry the specific amino acid. Now the messenger RNA in prokaryotes, the messenger RNA is almost the same, and it is polycistronic. More than one genes, more than one coding region, they are present in the prokaryotic messenger RNA, so as to synthesize more than one polypeptide chain. But in case of eukaryotic messenger RNA, the messenger RNA, primary transcript, it is elongated, and further modification take place to add the 3' poly A tail, 5' cap, and 5' cap of the 7-methyl guanosine. And along with it is monocistronic. Monocistronic mean only one gene for a single polypeptide, or one coding region for a single polypeptide chain present in the eukaryotic messenger RNA. The eukaryotic messenger RNA, as well as in prokaryotes, the messenger RNA provides the template for the protein synthesis. So the link between the polypeptide chain as well as the nucleotide sequence on the DNA is the messenger RNA. In case of eukaryotic transcription, and Also in the prokaryotes, the DNA-directed RNA synthesis takes place, and it comprises the initiation, elongation, and termination. Initiation involves the promoter region both in prokaryotes and eukaryotes. The prokaryotic promoter region, they are recognized by the sigma factors, while in case of eukaryotes, more complicated, more complex phenomena occur in eukaryotes, and it involves the transcriptional factors. These transcriptional factors, they are known as the trans-acting factors because they are synthesized from a gene, then goes to the cytosol from where the translation takes place, and further post-translation modification of these proteins. And now these proteins, they act as a translational factors for the other gene. Similarly, initiation, it is the recognition of their specific promoter region both in prokaryotes as well as in eukaryotes. However, the elongation process, it undergoes the elongation of or the strengthening or the synthesis of the um, RNA. In the elongation, the polymerase always read the template DNA in a 3' to 5' direction. Always read the nucleotide sequence on our DNA by the RNA polymerase enzyme in 3' to 5' direction, but it has the ability to synthesize a nascent RNA, a ribonucleic acid polymer, in 5' to 3' direction. It means that the the nucleoside triphosphate, it will be added at the 3' end of the newly synthesized RNA with the release of the pyrophosphate. Nucleoside monophosphate, actually the nucleoside triphosphate is the substrate for the addition of a new nucleotide at the 3' end. The nucleoside monophosphate will be added with the release of pyrophosphate. This growing RNA always take place in 5' to 3' direction, and this is the elongation phase until the termination phase take place. When there is a termination sequence present in eukaryotes, or the termination sequence present in prokaryotes, which give rise to stem and loop structure, or the row dependent termination. In case of prokaryotes, termination may be a row dependent or may be a row independent. But in case of eukaryotes, this termination, it involves the termination sequence, and thus the separation of this RNA from the DNA take place after this reading of the termination sequence. Both in prokaryotes and eukaryotes, the newly synthesized transcript of RNA undergoes various modifications, which is known as the post-transcriptional modification. Instead or except in messenger RNA, the messenger RNA of most, or you can say that in all of the prokaryotes is the same as the nascent messenger RNA. However, in eukaryotes, it undergoes the various complicated post-transcriptional modification, which involve the 3' poly A tail, 5' capping, as well as the splicing reactions. The ribosomal RNA, in case of prokaryotes and eukaryotes, it undergoes the various cleavage as well as the trimming reactions. Trimming reaction take place by the exonucleases, while the cleavage by the endonucleases, and to give rise to the functional ribosomal RNA species. And similarly, in case of the transfer RNA, there is the cleavage, there is the of the terminal 5 prime as well as the 3 prime nucleotides, as well as the cleavage of the intron, cleavage at the 3 prime sides, the use uracil nucleotides, as well as the addition of the 3 prime CCA. This addition of 3 prime CCA is basically it involved for carrying the amino acid in uh, or bringing this amino acid, specific standard amino acid, towards the messenger RNA template. All of these are discussed previously in transcriptional. Um, uh, Now this is the different ways by which a prokaryotic transcription take place. If we revise or recall this one, that this is the RNA polymerase, which uses sigma factors, and for the identification, for the recognition of the different promoter region. These are the promoter region of the prokaryotic promoter region -35 sequence and the Pribno box, and sigma it involved for recognition of these promoter regions. But if promoter regions they are changed for a different gene, then sigma factors they are also changed. So this phenomenon is known as the sigma switching, very helpful for the recognition of those for different types of promoter region of different genes in case of prokaryotes. And this one is the elongation phase in prokaryotes. Along with there is a strengthening or the growing of the newly synthesized RNA transcript in 5 prime to 3 prime direction. As in both prokaryotes and eukaryotes, this transcript always grow in 5 prime to 3 prime direction, so it is directional. Similarly, in prokaryotes, the termination may be a row independent to form a stem and loop structure, or maybe it is row dependent, which involve the protein so as to terminate the transcription. In eukaryotes, a complex promoter region present, such as the proximal promoter region, distal promoter region, as well as the core promoter region. Core promoter region basically in some genes contain the Tata box or Hodgkin's box, and but in most of the cases, instead of this box, there is another promoter region, that is the downstream downstream promoter element, or there is the initiator box in most of the genes. Different transcriptional factors they attach to their specific promoter region, specific elements. These elements may be response elements. Here, these response elements, there are thousands of base pair away from the transcriptional start site, and they are responsible for the initiation of gene expression. Here is the binding of the general transcription factors. Transcriptional factors, they are of two types, maybe a general transcription factors, maybe a specific transcription factors, and binding of these two types of transcriptional factors to form the transcriptional complex. Here is the binding of the general transcription factors on their specific elements on the DNA, so as to express the so as to start the transcription in eukaryote. So very complicated process which involves the remodeling of the chromatin and the acetylation reaction, so as to the ease for the transcriptional factors to their specific promoter region in eukaryotes take place. Now, next is the protein synthesis and the genetic form of code, and this is our first lecture on the protein synthesis and genetic code. The genetic information always travel from DNA to form the protein. So, this is the central dogma. A central dogma involve the synthesis of messenger RNA, then translation process of this messenger RNA take place. And various components also help for this translational process. It involves the tRNA and ribosomal RNA. Central dogma also involves the replication process from DNA to DNA during the cell division. So this one is the flow of information from DNA to RNA and then into the polypeptide chain. This process of protein synthesis required the genetic code. Genetic code, it is actually a dictionary or table which is required for the synthesis of a polypeptide chain. And post-translational modification may occur or mostly occur in most of the proteins so as this protein fold properly, and there may be equivalent modifications to activate or inactivate the protein. However, there are mutations on the nucleotide sequence present on the DNA which lead to the mutant polypeptide synthesis, and this mutant polypeptide synthesis may cause the disease state and even death of the organisms. And the signals present on the proteins which are targeted to intra or extracellular destination, all of these signals, they may be added after the protein synthesis. So nascent protein undergo to the post-translational modification to perform their proper functioning. Now the genetic code, it is the dictionary which provide uh, template, which provide uh, the you can say translate how these nucleotides they go into the formation of a polypeptide chain. A codon, it is actually comprises the three nucleotides which are present on the messenger RNA. In a codon, instead of thymine, uracil is present. So uracil replaces thymine on messenger RNA. The sequence of three nucleotides, 8 form a codon on the messenger RNA. The codon, there are the 64 codons present in our genetic code, and on these 64 codons, there are the one is the initiation codon, AUG is the initiation codon, three are the stop codon. AUG is the initiation codon and also it codes for the methionine. So this initiation codon, when initially present or start, it is present at the start of the messenger RNA, and maybe it is present in between of this messenger RNA, and it involved for the addition of methionine. And there are three codons which are known as the stop codon for the termination of the translational process. This one is the stop codon. UAG, UGA and UAA are the stop codon.